In this video, we will show you how to simulate gateway cloning with a single fragment on Mendelgen. Gateway cloning takes place through two recombination reactions, the BP reaction and the LR reaction. The BP reaction takes place between a vector containing the insert of interest flanked by @B sites and a donor vector containing @P sites. Recombination between the @B sites and the @P sites produces an entry clone containing the insert of interest flanked by @L sites. The LR reaction then takes place using this entry clone with the insert of interest flanked by the @L sites and a destination vector containing @R sites. Recombination between the @L sites and the @R sites then produces the final vector called the expression clone with the insert of interest flanked by @B sites. To begin the gateway cloning simulation, we open the Mendelgen dashboard. And from here, we select the vector containing our insert of interest. This vector does not contain the @B sites yet. They will be added later. So in this example, I would like to use the vector pmd2.g. After clicking on the vector, it will open the editor, and we will see the vector on the left in a circular map and on the right in a sequence map. Now, to start the cloning simulation, I will go and click on the cloning icon above. When it's clicked, it will show a drop-down listing all the cloning techniques that are supported within Mendelgen. I will click on Gateway Cloning. This opens a cloning tab to the right that will clearly outline all the steps that need to be completed in order to perform the cloning simulation. In the first step, we must identify an area on the vector that we would like to treat as the insert. This can be done either by clicking on a feature, which will highlight an area, or clicking in an area and then dragging the mouse and then releasing. This can be done either in the circular map on the left, or you can switch over to the sequence map and perform the same actions, such as clicking simply here, or clicking, dragging, and releasing. Okay, so now in this example, I'm going to select the feature VSVG simply because it's large and we'll be able to easily see it in the expression vector at the end. On the next step, we need to specify the orientation that we would like the insert to be in the final expression vector. Now, the forward here means the orientation as shown in the viewer, where reverse will be the opposite direction. So, as we can see with the pink arrow here, VSVG is already in a forward orientation. So forward, we'll keep it like this in the final expression vector. However, if we were to switch it to reverse, then VSVG would be in the opposite orientation. In this example, I would like to keep the orientation forward, so I'll switch back, and then I'll click on Create Insert. And the insert will then be shown below Insert Created. Now, something that you can't see here, but has taken place in the back, is that when you clicked Create Insert, the @B sites that we specified earlier have been added flanking this insert region. Now, as we described, these @B sites are required for the BP reaction with the donor vector. Now, this takes us to the next step, where we have to choose our donor vector. Then we can click on the drop-down below, choose a donor vector, and it will show all the vectors we have uploaded into our library on Mendelgen. In this example, I'm going to select the vector pDonor223. The vector will then be shown below donor vector and highlighted in blue. This highlighting in blue lets us know that if we click on the vector, it will open in a new editor. Remember, our donor vector contains the @P sites. So I'm going to click the vector and open it in a new editor to ensure that the @P sites exist. Now, having used this vector before, I know that this feature here is an @P site, and this feature here is also an @P site. So then we know that the pDonor vector can be used as the donor vector. Okay, and I'll switch back to my cloning simulation tab. And we'll remember that between the insert and the donor vector, we have the BP reaction, where the @B sites in the insert will go through a combination with the @P sites and the donor vector, creating an entry clone that has the @L sites flanking the insert of interest. In the final step, we will choose the destination vector, which contains the @R sites that will go through recombination with the @L sites on the entry clone. Now, when we click the drop-down below Choose a Destination Vector, just as above, we will see all the vectors that we have preloaded in Mendelgen in our library. In this example, I'm going to choose the vector pdest10. Now, same as above, we see the vectors shown below in blue, and if we were to click on it, it would open in a new editor, and we could ensure that the @R sites exist. Now, I've already used this vector before, so I know that they're there. Now we can just go ahead and click clone. So the gateway cloning simulation has taken place, and as we can see, our sequence of interest is right over here, with the VSVG feature and the pink arrow still pointing forward, so we know the orientation was maintained. This concludes our video on gateway cloning simulation. Please check our other tutorial videos to see simulations for other cloning techniques.